The NMDA receptor is important in learning and memory. We're going to go over what it does in terms of normal physiological function and when it goes wrong in diseases such as Alzheimer's. First, we're going to go over how a normal neuron functions, and then we're going to get started onto the NMDA receptor because it's a little bit different, its signaling as opposed to just typical receptor signaling. So here we have two neurons, neuron 1 and neuron 2. The first neuron has something known as a vesicle, which has neurotransmitters inside of it. So right here, right? These circles are the neurotransmitters. This big circle is the vesicle. Whenever something known as an action potential, which is an electrical impulse, reaches the nerve terminal, so this is the nerve terminal, it causes the change in a voltage, which opens something known as a voltage-gated calcium channel. So this channel right here is closed, let's say, but when the change in the voltage is sensed in the membrane, it allows calcium to enter. So it's this one actually, sorry. And once calcium enters, it enables for the release of these neurotransmitters into the synapse, which is the space in between two neurons. Now the neurotransmitters can diffuse and bind to the receptors of the next neuron and have activity there. Some neurotransmitters are excitatory, such as glutamate. Some are inhibitory, such as GABA. GABA causes an opening of a chloride channel, and chloride has a minus charge, meaning that it makes the cell more negative and less likely to fire an action potential. Glutamate causes an increase in positive ions, meaning it makes the cell less negative and therefore more excitable, all right? More likely to fire an action potential. So the NMDA receptor right here is a glutamatergic receptor. It has other ligands that can bind to it, such as glycine, as you can see right here. And there's also an Ampa receptor. The major differences here you need to notice are that the NMDA receptor is blocked by magnesium. All right, and the Ampa receptor is not blocked by magnesium. The reason why this is important is because you will need the magnesium to be displaced in order for the NMDA receptor to become activated. And again, this is a very important receptor for learning and memory. We'll see that because in Alzheimer's, some of the ways the neurodegeneration takes place is because of too much glutamate activity that causes neuronal loss via something known as apoptosis via calcium signaling. All right? Apoptosis is one of the mechanisms of neuronal death. But let's get started. So first thing that happens is you have a neurotransmitter that binds to this Ampa receptor. Again, Ampa is permeable to sodium, also potassium, but importantly, in terms of understanding what happens here, the sodium enters the cell, right? And it goes in and it displaces the magnesium. So this magnesium is no longer in the way. And once that magnesium is out of the way, another glutamate molecule can now bind to the NMDA receptor. Now, more sodium can enter the cell, as well as calcium. Now, the calcium influx can cause cellular change via signaling cascades that can cause the increase in the number of more Ampa receptors. What does this mean? This means that you can have a stronger synapse because you have more receptors. That means if you have 3 neurotransmitters, right? Let's say we have 1, 2, 3, and before you had only 3 receptors, right? And some of the neurotransmitter can diffuse away or get taken up by a glial cell. But now you have, let's say, 4. The probability of binding to a receptor increases, and therefore the synapse is known to be strengthened. So synapses can be strengthened or weakened based on their activity. So this is a short-term change to the synapse. There's long-term changes as well, such as the growth in things known as dendritic spines, and that's a later process. That involves the changes in gene expression via CREB, which is cyclic AMP response binding element, which is a transcription factor, which is just something that affects gene transcription and leads to changes in basically morphology of the dendritic spines, which are just the protrusions from the dendrites. All right. So one more time, we have Nmda receptor blocked by magnesium. So basically, it's not going to let anything go through, right? If anything, it's going to be very minimal, right? But then we have an Ampa receptor here in blue that is permeable because it's not blocked by anything, right? So let's say glutamate binds. Glutamate is excitatory, so we know it's going to be positive ions that are going to enter. In this case, sodium. So sodium comes in and goes and kicks off the magnesium. Now calcium can enter. And when calcium enters, it causes cellular change, which leads to these receptors being put onto the surface, strengthening the synapse. Now you can basically use less neurotransmitter to get the same effect. So the synapse is strengthened. But what I was going to say, actually, sorry, one more time. Now that you can bind glutamate without a block, it's even stronger because calcium can enter as well as sodium. So you have more positive ions. So 2 plus is more than just 1 plus because sodium has a plus 1 charge, calcium has a plus 2 charge. So there's even more ions that can flow through that as opposed to just what can flow through the Ampa receptor. All right. So what else did I forget to mention? Oh, here, this reuptake receptor is one of the mechanisms of which neurotransmitters are cleared from the synapse. So you have a signal and you don't want the signal to be infinite because the neuron can become basically overexcited to the point where it dies. It's known as excitocytotoxicity, and we'll see that in a little bit. So you need ways for these neurotransmitters to get removed from the synapse, and one of the ways is to bring them back into the receptor by reuptake. Usually a transporter, like dopamine transporter has its own called DAT. Serotonin has its own known as SERT, serotonin reuptake transporter, and so on. And norepinephrine has its own as well. And there's probably general ones also, but don't quote me on that one. But there is definitely general ways of eliminating neurotransmitters from the synapse via, let's say, monoamine oxidase, which is a general enzyme that gets rid of monoamine via oxidation. Monoamines are a class of neurotransmitters which have one amino group on them. I'll take a sip of coffee and then I'm going to summarize one more time. Nmda receptor is blocked by magnesium. Whenever sodium enters via Ampa receptor, it's able to displace magnesium. It's kind of like, let's say your front yard door is closed. You go through the backyard door and you come back from the inside of the house and you open the front yard door. Now you can enter to the front. More people can enter the house because they can take the backyard door as well as the front yard door. All right. So calcium can enter, sodium can enter, and calcium is going to cause cellular change, such as the receptors being put onto the surface, as well as the growth of dendritic spines in the later phases. If you have any questions, always feel free to comment. Clinical relevance. There's drugs that target this receptor, such as ketamine. There's recently been a drug known as Spravato that's been approved. It's Esketamine, which is basically an enantiomer, which is just a version of ketamine that has a particular 3D shape. And let's not get too much into that. That's like organic chemistry, but it's like left hand and right hand kind of thing. All right. They might have different functions because there's different receptors, and some receptors, which is basically where the drug binds to, can have basically shapes that are more favored towards one shape than another shape. Right? All right, let's continue. PCP is a street drug. That also blocks NMDA receptor. If you look at the structure, it's kind of similar to ketamine, but obviously it's a much different drug. Memantine is used for Alzheimer's disease in later stages. It is an NMDA channel blocker, as well as magnesium as a supplement. What do all these drugs have in common? They're NMDA receptor antagonists. Antagonists are drugs that basically block, and agonists are drugs that basically do the opposite of blocking, which is enable. All right. Here I put not activated. I forgot to mention this earlier. It becomes activated whenever the magnesium gets removed, just so you don't confuse that. All right. So these are all NMDA receptor antagonists, right? And basically what that just means is that they basically block the activity of that receptor. An example of an agonist is glutamate, because glutamate allows for the activity of that receptor, enabling the increase in the amount of sodium that enters, the amount of calcium that enters whenever the magnesium block is removed. And here there's another image of how it can be represented. This is the membrane portion, and this is the intracellular portion, this is the extracellular portion, and they're just this is just a different representation of the structure. Now let's talk about how ketamine works. So ketamine blocks an NMDA receptor, but it's used for depression. How does that work? Well, it's interesting because you think that someone who's depressed does not want to have their NMDA receptor inhibited, right? Because that means that their neurons are less active. But this is an interesting scenario because the way it works is via double inhibition. So double inhibition means excitation in this particular case, because you have something known as a GABAergic neuron. GABA is an inhibitory neurotransmitter, meaning that it decreases neuronal activity via the opening of a chloride channel. Chloride has a minus charge, so it causes the cell to be more negative, and by blocking the release of GABA, you're basically removing the brake off of the neuron and causing the neuron to fire. So this is the neuron of interest, let's say, right? And we want this neuron to fire. But it gets blocked by this GABAergic neuron when this GABAergic neuron is active. Now, ketamine acts on the NMDA receptor that's located on this GABAergic neuron and inhibits that GABAergic neuron, and thereby blocking the blocker, causing the activity to increase as a result, which basically results in some of the therapeutic benefits of ketamine in terms of treatment-resistant depression, as well as I forgot the other kind of depression. All right, you can Google the indications for Spravato. All right. Now let's continue. Anything else? No, I think I talked about all this. I put these notes here in case I went too fast. Memantine. How does memantine work? This is a structure of memantine right here, and I represented it here with a diamond. And this diamond looks like glutamate. Glutamate is a diamond in red, just to show you that basically the idea is that it competes for the same site, which leads to less activity on this NMDA receptor because glutamate is not able to bind. Why is memantine used in Alzheimer's disease? It's because in Alzheimer's disease there's too much glutamatergic activity, meaning too much glutamate is acting on the NMDA receptor and causing too much calcium to enter, and too much calcium is excitocytotoxic, meaning it causes cell death via the activation of cellular enzymes that degrade the cell, as well as basically the whole apoptosis pathway, the intrinsic pathway, which is a mechanism where you have in your mitochondria the release of something known as cytochrome C, leading to the activation of something known as caspase 9, I believe. Anyways, caspases, which lead to a caspase cascade, which lead to cell death via chewing up proteins and whatnot. So big picture is just that calcium levels need to be kept low. That's why there's something known as the endoplasmic reticulum that has calcium stores inside of the cell. So there's ways that calcium is basically kept low, and one of the ways is it's kept inside of the ER, as well as binding proteins that bind to calcium, as well as pumps that pump calcium out of the cell. So there's various mechanisms to keep calcium very low, because too much calcium is cytotoxic. So anytime you see that there's various mechanisms for something, that usually tells you that it's important, evolutionarily speaking. All right. Now, what is Alzheimer's disease? Alzheimer's disease is a neurodegenerative disease. It's the most common cause of dementia. And basically, what is dementia? It's cognitive impairment that basically affects daily living, everyday activities, or whatever. Sorry. And they diagnose it via cognitive tests such as MoCA. There's other clinical assessments and whatnot that are available, but to my knowledge, the MoCA is probably the most common expertise in all of this stuff. So there's probably other things like scans that are done, but I only put here just the big picture stuff. And then there's risk factors such as ApoE alleles. An allele is just a version of a particular gene. So certain versions of this gene can put you at a greater risk of developing the disease. Age is a risk factor, as with many diseases, and the environment as well. So in particular, head injuries are going to be something to watch out for. Risk factors are not causal, right? Just to keep that, just to make sure that that's clear. What can you do to possibly help? There's something known as cognitive reserves, which is basically analogous to putting on sunscreen. So let's say if you're going to get sunburned, by adding sunscreen, you take longer to get sunburned. So by socializing and exercising and eating right, you increase these cognitive reserves, which basically will delay the onset of symptoms or whatnot, if that makes any sense. Now this is basically the beta amyloid formation, a schematic diagram of how it happens. So the beta amyloid is proposed to be one of the ways that Alzheimer's disease is generated, basically. How are the neurons killed in Alzheimer's disease? Is via these plaques being formed. And basically, the amyloid precursor protein here, APP, this tube right here, is a normal protein, right? But the issue is it can be cut in an abnormal way, which can lead to the formation of an abnormal protein. So the good way would lead to no problems, right? So alpha secretase, here I put 1 prime, that's the normal cut. Let's say it cuts right here, and it leaves, let's say, this, and it leaves that. One is extracellular, the other one is embedded inside of the cell membrane. This is a cell membrane, it's embedded inside of it. And then it gets cut by something known as gamma secretase and leaves another portion, right? And that does nothing, I guess. Nobody's sure exactly what that part. Apparently it's confusing. But you form a product that doesn't do anything. Now, beta secretase can cut it in a way that generates a portion that stays embedded in the cell membrane, as well as generating A beta. The portion that stays inside of the cell membrane gets cut by gamma secretase and goes into the nucleus and can cause changes in gene expression. All right? The A beta can obviously go ahead and clump together and form these plaques. These plaques basically will damage neurons and whatnot, cause inflammation, and block neuronal communication. And we'll look at that in a more macroscopic view. All right? So the big picture here is you have 2 enzymes. One cuts the amyloid precursor protein in a bad way. The other one cuts it in a good way. And basically, the products, the result, will either cause problems or not cause problems. There's also tau, which is also implicated in Alzheimer's disease. So there's tau hypothesis, and there's obviously maybe tau and amyloid acting together. There's various different things that are taking place in Alzheimer's disease. So there might not just be one cause. But the way tau might be involved is the following. So your cells, let's say this is a neuron, let's say it gets a signal from these dendrites, which is the antennas. It has to basically propagate that signal down the axon onto the nerve terminals and cause neurotransmitters to be released. Now, how do these neurotransmitters get to this very end right here? They use microtubules as the highways. This Pac-Man right here I didn't talk about earlier is something known as kinesin or dynein. Kinesin is the one that we're talking about here because we're going towards the nerve terminals. So we'll ignore dynein for now. In order for this Pac-Man to be able to walk across this microtubule, the microtubule needs to be stable, and it is stabilized by tau. Now, if tau gets phosphorylated with this lightning strike symbol, it disassembles from the microtubules. Now the microtubules are no longer stable, and they disassemble into individual monomers of alpha and beta tubulin. And now the neurons are not able to basically receive these neurotransmitters, and they are basically unstable. So now the neurotransmitters are basically not able to be carried along the microtubules, so communication is basically impacted in that sense. What amyloid does, amyloid blocks neurotransmitter communication. It causes inflammation via the activation of microglia. Microglia are your immune cells inside of the brain. They chew up things, basically. But too much activity of microglia can also be damaging because they produce reactive oxygen species. Amyloid also, by causing all this inflammation, eventually leads to mitochondrial issues as well, which basically also causes damage, causes a bunch of problems. That's the big picture. It'll be impossible to go through all the stuff that it does, but basically, tau is important for supporting microtubules. If it's phosphorylated, let's say by this lightning bolt, it doesn't stabilize these microtubules. Now, the Pac-Man can't carry the neurotransmitters to the end of the nerve terminals, and now the communication is messed up. And yeah, there's multiple drugs that are used to treat it, depending on the severity. Another drug that I did not mention, I talked about Memantine, but there's cholinesterase inhibitors. These block the activity of acetylcholinesterase, which is an enzyme that degrades acetylcholine, a neurotransmitter that's important in cognition. And so by blocking this enzyme, you have more neurotransmitter available in the synapse, so there could be better communication. And remember, we talked about earlier how amyloid can block these communication. And these circles right here are your neurotransmitters that I'm representing here. And again, the Pac-Man, which would be the kinesin, is carrying those neurotransmitters onto the microtubules to the nerve terminal. All right. And just as anything else, there's always exceptions. So for some reason, there are individuals who have Alzheimer's brains, but do not have the Alzheimer's phenotype, meaning that they're not demented, so they don't have dementia. So this is like post-mortem histological sections, I believe, that they look at, histological sections, and they look at the brain and they say, what's going on here? This person did not have dementia when they were alive, but their brain looks like someone who should have dementia. So just like anything else, there's always exceptions. So summary of the mechanism of Alzheimer's in the amyloid pathway. Amyloid precursor protein is a normal protein that can be incorrectly cut and cause amyloid plaques and tangles. They cause problems when they accumulate. Phosphorylated tau causes neurofibrillary tangles. Neurons have roads on them, microtubules, where the neurotransmitters are carried, and the tau is like the glue. When it gets a certain chemical modification, phosphorylation, the glue is no longer able to hold the road together. It adheres to itself and forms these tangles. Overall, altered neuronal signaling inside and outside of the neuron leads to loss of neurons due to these disturbances. Neuronal losses examples are reactive oxygen species generation via activation of microglia, caspase activation leading to apoptosis. Thank you for listening, and if you have any questions, please send them in, and don't forget to please subscribe.